Hello, and welcome to today's webinar, Best Practices for Spirometry and ALS Clinical Trials. Before we begin, I'd like to review some key audience tips to give you the best webcast experience. Today's webcast is being streamed through your device, so there is no dial-in number today. Please check your speaker volume to ensure it is turned on. Also, for the best webcast experience, please use Google Chrome. Be sure to enable Adobe Flash, the media player, and we recommend closing all windows except for this browser. To change the size of the video feed and the presentation on your screen, drag the slide bar at the bottom of your screen to the left or right to adjust your screen size. Your control panel is on the bottom sidebar on your screen. The bottom red leave button is how you will disconnect at the end of the webinar. If you have any technical difficulties, please use the chat box. We have relevant resources available for you today. You can access and download these resources within the chat box. Please provide your feedback about today's webinar via the survey immediately following the broadcast. Finally, and perhaps most important, is the Q&A icon where you can submit questions to the presenters at any time during the webinar. Please use the window for presenters and the moderator chat for technical difficulties. If we are unable to answer your question during a webinar, we will follow up with you separately. So please do not hesitate to ask any questions. If you would like a copy of today's presentation, you can send a request through the moderator chat or later to marketing@ert.com. Finally, an on-demand version of today's webinar will be sent to you shortly after the webinar. And now let's get started. My name is Dawn Patterson. I'm the Director of Respiratory Solutions covering the East Coast. I am a respiratory therapist and have been for 33 years, and I previously managed a respiratory overlay department at ERT. I just started my 10th year. Joining me today is Dr. Kai B., founder and medical director of INSEF Respiratory Research Institute, Wiesbaden, Germany, where he has designed and conducted more than 100 clinical trials in asthma, COPD, and allergies, acting as principal or coordinating investigator. Dr. B is the author of the popular international nonfiction book, The Breathtaking World of the Lung, published by Random House in October 2018 in multiple countries and languages globally, as well as many other publications. Kevin McCarthy has been working in the field of lung function testing since 1973. He was the Pulmonary Function Laboratories Manager at Cleveland Clinic Health System for nearly 40 years, retiring in 2016, and was also a member of the ATS-ERS task force that updated the standards for spirometry in 2019. Kevin currently is the Director of Respiratory Solutions, my counterpart, covering the West Coast. In the webinar, learn how to improve the success rate in designing your ALS clinical trials. You will hear from industry's leaders as they discuss the inherent challenges in performing respiratory tests with ALS patients. The discussions will focus on how to best provide as much comfort to the patient while reducing data variability and improving quality of data capture and regulatory requirements for reporting clinical trials. Dr. B, can you tell us what is ALS and what are the causes? How long do people live with ALS?
Thank you, Dawn, and welcome to this webinar. ALS is, as we know, a neurodegenerative disease. So in simple words, we see a progressive loss of functioning of motor neurons, both in the brain but also in the spinal cord. So these motor neurons that control the skeletal muscle, which means that patients, as disease progresses, will have a progressive loss of muscle function. And that can present firstly in the limbs, extremities, but it can also, as a so-called bulbar form, first manifest with issues in swallowing and speaking. It's a disease that manifests at the time around 50 years and is usually progressive and there's no cure for it. So most people probably will die within 3 to 5 years after diagnosis.
Would you suggest subjects, patients use a BiPAP prior to your visit to collect spirometry? Will this misrepresent the results?
Well, many patients with progressive disease eventually have respiratory failure and therefore they need ventilatory support. So in these cases, depending on how long the patient is using the machine, which is very variable, I would say it should be a recommendation that they use the machine as long as possible. And then, of course, if you want to try to measure spirometry, you need to standardize it in a way. How long are they off the machine before you actually perform the measurement? But I would say it's a case-by-case discussion.
Thank you. Thank you. Dr. B, when the respiratory muscles are affected in ALS, how does this impact the patient? What are their symptoms?
Yeah, here's where the pulmonologist actually comes in, because in the end, it's still a neurological disease, and I'm a pulmonologist. However, as the disease affects respiratory muscles, both the in and the expiratory muscles and the diaphragm particularly, patients will develop respiratory symptoms. And that can be anything like daytime fatigue because they have desaturation of oxygen at nighttime, to dyspnea. Dyspnea both in the lying setting, also in the sitting setting. Quite often, earlier it starts with dyspnea in the lying setting because the respiratory mechanics makes it more difficult to breathe in the lying situation. So that's the typical respiratory symptoms. Then you can have consequences of the bulbar symptoms, which is aspiration, disturbance of swallowing. So these patients get respiratory infections because they also have reduced respiratory clearance of secretions. So that's the second problem that we face.
Okay. All right. So looking at the screen, you'll see supine positions. Do you discuss your patient's sleeping position and if they have issues sleeping? Can we discuss the issues with supine?
Well, clearly, supine dyspnea, so dyspnea in the lying, is something that comes earlier than sitting dyspnea, as I discussed with the altered respiratory mechanics. So it's very important to discuss the question of dyspnea or discomfort with breathing in the lying position with your patients.
Thank you. Thank you. Okay, so let's talk about spirometry. Kevin, we know vital capacity can reflect the impact of ALS on the respiratory muscles, but what exactly will it tell us?
So it's exactly that. That is, it's sort of an indirect assessment of how the disease is impacting the respiratory muscles. And when you think about the vital capacity maneuver, it involves both inspiratory and expiratory muscles. And so those could be sort of selectively involved, meaning just inspiratory, just expiratory, or both. And so it's a bit unpredictable what you're going to see when you try to do spirometry. And in fact, in the early stages, when respiratory muscle involvement is minimal, you may see a fairly well-preserved vital capacity. That is, when compared to the sort of broad range of normal predicted for that subject, it may not raise any flags. And as we were looking at that slide there, the sitting and supine, as Dr. B mentioned, there was the abdominal contents in the supine position sort of represent a challenge test for the diaphragm function, as it were. And so you can sort of tease out the impact of the inspiratory muscles by looking at the difference between the upright spirometry, the vital capacity measured upright, and the vital capacity measured when the subject is laying flat. So it's, again, you don't know what you're going to get until you make the measurement. And rarely is that the first presenting symptom that a patient with ALS will have. But it eventually does usually become a complaint that they're short of breath, and usually, as Dr. B said, it's when they usually start complaining about, when I lay down to go to sleep, man, I just don't feel right.
Right. That's right. Thank you, Kevin. Dr. B, how important is this information for clinical trials?
Well, as Kevin said and pointed out, it is a very important test to perform for diagnostic purposes to detect involvement of respiratory muscles, the performance of vital capacity maneuver. However, what is important in the course of the disease or in clinical trials is the question whether we can also use volumes like vital capacity as an outcome parameter. So do these factors act as a trusted biomarker for outcomes? And that is, in fact, true. We know that the decrease in vital capacity is associated with general worsening function, and it's indicative of the need to require ventilatory support, and it's also a predictor of mortality. So vital capacity is useful in diagnostic settings, but even more so as an outcome parameter in clinical trials.
Great, thank you. Thank you both. Okay, so let's discuss a few things on how to collect data from ALS patients. Now, there are always several discussions with the client on what is the best way to capture the effort without losing a good seal. Is it the mouthpiece or is it the mask? Now, my personal experience, I've used both the mask and the snorkel mouthpiece. And what I found is that with the mask, it's difficult to maintain a good seal with one hand. There are 3 points on the mask that will require both hands to ensure a good seal. The other issue that I found with the mask is that when a patient has a lot of saliva, you definitely have a difficult time, making sure that there's a good seal. So it's just so much easier and more comfortable for the patient to use the snorkel mouthpiece. So, Dr. B, what are your thoughts on that?
Well, I would agree with you, Dawn. Of course, it's always an individual decision. I mean, you really have to take the best fit. But in general speaking, I would agree with you that the snorkel mouthpiece is something that is easier to handle and probably produces better results than using a mask, according to the problems that you just illustrated. But that's my personal opinion. And of course, I've heard yours, and maybe Kevin has a different one on that. So I'd like to hear Kevin's viewpoint on that.
Certainly. I am in total agreement with you. That is, having spent a lot of time, let me just say that when I was working at Cleveland Clinic, we considered the mask to be sort of a last resort and a relatively unsatisfying last resort. We ended up having to identify a good selection of masks to accommodate the architecture of all the different types of faces that we could be testing. And as you say, Dawn, those 3 key points, bridge of the nose at the side of the corner and the fit down there makes it difficult. Saliva makes it difficult, but I would say that saliva is something that everybody doing these measurements should be prepared to deal with. You should always be gloved and have a washcloth or something to help wipe the patient's mouth or chin. But I would definitely cast a solid vote in favor of the snorkel mouthpiece. It's no question that you can assess the leaks much, much better, and you tend to get more repeatable data with fewer efforts.
Okay, great. So it sounds like the snorkel got the thumbs up, right? So, so now let's discuss the parameter. All right, FVC versus SVC. I'd like to get both of your opinions. Kevin?
Sure. And I should say that, there's not complete agreement in the pulmonary function testing world about what the definitions are for a slow vital capacity and a forced vital capacity. But I would say that what we're looking for here is vital capacity. And it comes down to what's going to get you the best performance. And I would say from experience that if you take it too slow, that's not necessarily going to work in your advantage. You might say, well, forced, that's going to be hard for the patient to do. True enough. And I would say that this is an instance where we might want to sort of disregard our notion of what constitutes a forced vital capacity, because we're really, in most cases, we're not really interested in the FEV1. We're interested in that total volume. So a rapid inspiration with continued coaching to be sure that you're at full inflation, I think, is important. Rapid, I think, tends to be easier for most patients to give it a great big effort that it's going to take them very, very close to full inflation. And then starting with sort of, okay, let it out, now push, you can say blast, but if they don't blast it out, okay, maybe they can't. Maybe the expiratory muscles are going to prevent that from happening. So don't be too concerned about that. What you're looking for is sustained expiration. So again, it may not really be fast would be okay. It's likely not to be best performance if you say, very slowly, where they're trying to hold back on it, but just have them try to sustain contraction of their belly muscles to get you a good long expiration is likely the best strategy that you're going to get.
Great, thanks. Dr. B?
Yeah, I would totally agree with what Kevin said. I think it's very important that we do not mix up a terminology in this case. So when talking about FVC is not what we're looking at in asthma or COPD, where we are really looking for maximum flow as an important quality criterion. And the point really is that we need to have an as accurate or as maximal as possible vital capacity. And the question is, how do we get the patient to achieve that? And I totally agree that doing it in a forced manner, or let's say in a supported blast, if you like, is probably the better way than doing it slow, because slowliness in this case really introduces variability. And that's something that we don't want to have in this situation.
Wonderful. Great point. Thank you. Thank you both. Okay. So we got the snorkel. We just discussed the parameter. Now let's move on to coaching the patient. All right. So we're at the site and the patient is sitting in front of you. How do you coach a patient with ALS? What do we need to take into consideration?
I can take this if you'd like. Yeah.
Go for it, Kev.
So I think first and foremost, you need to let the patient know that you're not going to rush this, because they're very usually full of anxiety about this procedure, and you want to make sure that they understand it. And what you need to keep in mind is that they may fatigue very, very quickly. So in the normal patient, when you're doing spirometry testing, it's okay to sort of correct as you go. And that's certainly true here as well. But we may have a very limited number of efforts where we're going to get maximum performance. So taking the time, having the patients to really say, all right, let's just talk. Let's talk about what it is we're going to do, so that when you get started, they have full understanding. They're not surprised by anything. They know what you're expecting of them. You've given them some tips on how best to perform that, and that they really are ready to go. And to that end, I would end by saying you really need to make sure that they understand that they have an element of control in the testing, that you're going to be questioning them between every effort, asking them, are you ready to give it 100%? And if they feel like they're not quite ready, it's okay. They should just let you know that, and you give them more time to rest, because if there's a patient population that may need it, this is it. And if you try to rush it, you're likely to hit the point of diminishing returns very, very quickly. And that's not our goal. Our goal is to get some numbers that really do represent what that patient's condition is at this time.
Great. Thank you. Dr. B, would you like to add anything?
No, I absolutely agree. Time is crucial. I mean, this is you really have to change your mindset with this type of patient. It's not this type of, okay, patient comes in, one replicate, second replicate, third replicate, repeatable, acceptable, okay, and now get out of my office. That's not right. The case here. Take time. It's like performing, let's say, a treadmill test with somebody who had a stroke or something. You have to take your time, and it's not about whatever getting 1, 2, 3 replicates in a reasonable order of time, but it's trying to make the patient comfortable and trying to get the best results that is possible together with the patient without intimidating him, and in the end, by making sure that you get the best possible result out of that test. And that's absolutely crucial.
Great. Thank you. Okay. So let's move on to evaluating the data. We collected it, and now we're going to evaluate it. Before a clinical trial starts, there's typically a discussion between the client and our OVA department to make sure that everyone is on the same page when it comes to data evaluation. Although there are guidelines suggesting how to evaluate the data, we need to take into consideration on what the indication is. Right? Sure, absolutely. So with ALS, what are we looking for when we're evaluating the data?
Great question. So having tested a lot and having worn an overreader's hat, I can tell you that this is one patient population where, again, we've already sort of covered this, that the total volume they exhale is really the key, the vital capacity. Even if it's not a perfect forced vital capacity, that's really what we're looking for. And so you really want to say, we're really not concerned about airway parameters like the FEV1. And so if it has a poor start of test, then we certainly don't want to throw it out, because we're interested more in end of test criteria and repeatability of that number. And so that's likely to be something that you would discuss right off the bat. And there was some, I can see it in your face. You're going to ask me about 2019 guidelines.
Maybe.
All right. So in the 2019 guidelines, one really nice advance, I think, was the direction to consider the quality of the FVC and the FEV1 separately. And so when you're asking my recommendations, that's a strong recommendation that you when you're looking at FVC, that you ignore the start of test criteria to a large degree. And you just say, I'm interested in getting that vital capacity.
Okay, great. Dr. B?
Yes, I would totally agree with Kevin. However, I would have one critical point here. I mean, I do agree that basically what we're looking at is vital capacity. And as we discussed with the 2019 guidelines, where you have this option, you have forced inspiratory vital capacity and you could force expiratory. So the question is, should I not just take the best out of these two? Just say, okay, maybe the inspiratory effort is just another opportunity for the patient to get his best maneuver. So scientifically and maybe clinically, I would totally agree. But having also had some experiences with regulators, which are more representative of the bureaucratic part of the universe, I just wonder whether there would be any difficulties in discussing with agencies when you get the best vital capacity from an expiratory versus an inspiratory maneuver. Because in theory, you could argue, well, okay, there are different muscles involved, so maybe the drug has a different or differential outcome on expiratory versus inspiratory force. So that is something that could come up. I'm not sure about that, but as I said, having had some experience with bureaucracy, this could in theory be the case. How would you respond to that, Kevin?
Thank you for bringing up what I think is probably one of the most important aspects of the 2019 guidelines in clinical trials, and that is verification that the subject really did start that forced exhalation from full inflation. And in that regard, I think that, I would be against using the demonstration of a larger FIVC to say, that's evidence that we need to throw out this forced vital capacity, because we've proven here by virtue of a larger FIVC that they really didn't start from full inflation. I would argue in this particular patient population, with the goal of testing being getting the largest vital capacity, that you should look at that larger FIVC as another opportunity to demonstrate the patient's largest vital capacity. And to your very valid point that, do regulators see it that way? I think that the 2019 Spirometry guidelines paper itself could be pointed to as evidence that a group of experts had the consensus that there really should be no difference between the forced vital capacity and the FIVC. That is, as you point out, different aspects of that maneuver involve different muscles, but there really should be no difference in maximum inflation before the start of a forced exhalation and maximum inflation at the end. If there is larger, in this case, I'd say that's great. That's another opportunity. We've essentially gotten two opportunities to produce a good quality vital capacity within the context of a single measurement effort. So thank you for bringing that up. I think it's a great point, and I'm sure that's one that's going to be on the minds of some of our listeners.
Great, thank you. So besides spirometry, there are other pieces of equipment out there that will measure the patient's muscle strength and their lung function. And I know that we have equipment that we can integrate into our spirometer. So looking at the slide, you'll see the muscle strength of the subject. Can we discuss this?
Sure.
It's our MIP and MAP, MIP and MAP and FOT.
Yeah. MIP and MEP have been used in clinical trials. They're really supposed to give you some direct indication of respiratory muscle strength, or in this case, weakness. They're not without their problems in terms of testing. Just as spirometry testing in this patient population is difficult, so is the assessment of respiratory muscle strength. And so use of those parameters as adjunct measurements, something that can help reinforce what might be a sort of an iffy kind of finding with respect to repeatability or quality in one test, might be pointed to again in the results of another measurement. And so that can be helpful.
Okay, great. Thank you. Dr. Bay, do you have any additions?
Yeah, I would agree with that. I mean, in terms of clinical outcome assessment in a clinical trial, there is, particularly in the milder forms of the disease, which is my understanding, no gold standard for monitoring respiratory involvement. So I think we need to think about additional measurements to establish vital capacity that could maybe first in an exploratory way, but maybe tell us something more about the progression of respiratory failure in these patients. And it's very helpful to use these. Well, technically, they are older tools. I mean, this is physiology that comes in part from the 60s and 70s. But we have been, in large parts of medicine, been so obsessed with molecular medicine and inflammation and T cells and stuff like that, that we sometimes forget about good old physiology. And these are beautiful tools to give you a nice idea of respiratory physiology, which is very important in these patients. And that could give us a better understanding of progression of respiratory failure.
Wonderful. Thank you. Okay, so we just discussed collecting spirometry at the site. So let's move on to collecting spirometry at home. Dr. B, what are some of the reasons spirometry is collected at home?
Well, there can be in general speaking, there can be different reasons for collecting data at home. First of all, you have the opportunity to collect more data. You can do it in a more frequent way without having the necessity to visit a site. That's one point. Then during the pandemic, for example, we had a situation where there were restrictions of access to sites, like with COVID-19. So that could also be, of course, attractive for doing home spirometry. And then finally, for many patients, can be quite burdensome to travel to sites. So this could be an alternative. If I think of ALS patients, usually they're quite sick. So for them, this can really be burdensome to travel to specialized trial sites, something like that. So these are the basic reasons why you would do that. One thing that is important for sponsors is that you shouldn't mix home spirometry let's say in a traditional asthma trial with home spirometry in an ALS trial. Because in asthma and COPD, that's totally different. There we strive that patients do the measurement more or less autonomously. So you give it in their hand, you train them, and then you get good quality data from the home measurement. That's different in ALS. Of course, in ALS, still, it takes a lot of effort. You need a lot of stuff, because that needs to be done in the same way, but in the home environment. So you need assistant staff, you need well-trained respiratory technicians and stuff like that. So it's different from, let's say, the traditional way of doing home spirometry. Nevertheless, I think it's feasible, it can be done. Mm-hmm. And of course, I would love to hear from your side whether you have any experience with these types of measurement in ALS patients.
Yeah, it's funny that you ask, because we actually just recently had a client reach out to find out some information on collecting good quality data from home from the ALS patients, because they were not able to travel to the site. So the one thing I explained right off the bat is that it is a team effort, and we all need to communicate so we're all on the same page. I mean, they really need to understand that. The team would include like the family members, the healthcare providers, and of course the site. The site may need to log in remotely to coach the patient in case the healthcare provider is unable to coach the subject. Or, I mean, if the healthcare provider was the one making sure that there was a good seal. So when the site remotes into the patient's device, they can actually coach that patient every single phase. So I was explaining this process to them, and they were like, oh, wow, that's really good. That's really nice. But we also have like outliers and quality data reports that the site can look into. Mm-hmm. So let's talk about the training. Everyone involved, they're trained on the equipment, they're trained on coaching, collecting, and evaluating the data. I mean, not only the site, it's the family member as well as the healthcare provider. And it's highly suggested, though, that the same technician to collect the data for all visits. But you know what? It may not always be the case. So that's why it's really important to make sure that everybody is trained. The other consideration was the environment. Does the house have enough room for a bed? So when we're collecting in the supine position, the patient needs to be comfortable. So, we know that some sites that it was always an issue. So you know what? The client overall, they were very happy that we were able to support a solution to ensure that we were able to collect good quality data for the trial. So it was a thumbs up. So they were very happy with that.
So it is possible.
It is possible. There are many, many ways that we could collect, data from home, whether it is with, our sensor, our iSpiro solution, or if it's with our regular spirometer. So there are several ways that we can collect data from home. So it's good. I really want to thank you both for your time to discuss how spirometry testing of patients with this disease can be challenging, not only for the patient, but also the operator. From this discussion today, it is apparent that proper training in patients are essential. And in addition to providing information about that full inflation part, that fourth phase specified in 2019 guidelines, it can provide a second chance within each effort to obtain the best quality data. And a huge, big thank you to our attendees for joining us today. We addressed a number of questions during our discussion, but if you have any remaining questions, feel free to submit them with the Q&A icon. For any question that we were not able to answer today, the team will follow up with you. Please take a moment to complete our survey and share your feedback on our webcast. Enjoy the rest of your day, and please stay safe.